project is, the big question is what's different about brain development in Down syndrome. And so what we are trying to do is using postmortem tissue and human stem cells to identify differences in how the brain forms in individuals with Down syndrome compared to those that are unaffected. And we're hoping to identify differences in the cells of the brain and differences in genes that are expressed, when those genes are expressed, and kind of genetic or molecular pathways that might be different. We are using what we call induced pluripotent stem cells. So these are cells that are generated from either skin cells or blood cells from anyone. In our case, we use skin cells from individuals with Down syndrome, and we reprogram them to behave like human embryonic stem cells. And what that means is we can grow them in the lab, in a culture dish, indefinitely, but more importantly, they can be coaxed to differentiate into any cell type in the body. And when they, in our case, we're differentiating them into brain cells, and as we do that, we can recapitulate or model the steps of brain development.
So we look at our cells every day. We want to make sure that any cells that are spontaneously differentiating, we remove those so that we have just great stem cells to work with to create our organoids with or neurons with. We want to feed them every day. They do not take weekends, so we are in here every single day and checking them and making sure that they look great. We also need to be very organized in what day we're switching different patterning molecules, what needs to be done with the cells, so usually keeping a calendar allows us to have this more organized so that we know what to do with our batches. When I was rotating with Anita, I was really shocked to find that despite the fact that Down syndrome is the most common genetic cause of cognitive disabilities, we really don't know how that extra copy of chromosome 21 leads to all of the anatomical and cognitive differences that we do see. So I think that with any science, the technology is going to be what limits what we can do. And at this point, with development in induced pluripotent stem cells, with these cortical organoids, and advances in transcriptomics, I think that this is a great time where we have all of the tools to start answering these questions as to why are we seeing these differences with an extra copy of chromosome 21.